Hi Ian, welcome back. This is the second of our videos about how substances move around in biological systems. We're talking about the process of osmosis, which is effectively the diffusion of water. Now, when you're looking at diagrams and you're thinking about what's going to happen in osmosis, you've got to think to yourself, the membrane surrounding cells in living things is partially permeable. In other words, some substances cannot pass through that membrane. So where these solute molecules would like to diffuse, they can't because they can't pass through the membrane. However, the water molecules can move through the membrane, and water is going to move from where there are more water molecules to where there are fewer water molecules. Sometimes that's a little bit confusing because the most amount of water is a dilute solution. So it sounds like osmosis is the opposite of diffusion when it isn't. It's just particles moving from where there's more of them here to where there's less of them here, and we're only talking about water. So in this diagram here, the water is going to move into this solution because there are more water molecules here than there are here. This is very dilute. There are lots of water molecules. They're going to move through the membrane and into the solution because there are more water molecules on this side than on this side. When we put plants in different strength solutions, normally for a plant, the contents of its cell will be more concentrated than the water that you're giving it to keep it alive. And so on the whole, water would move into the plant. So in this diagram, we're showing that water is going to move through the cell wall. It can move through the partially permeable cell membrane, and the water will fill up the contents of the vacuole, which is this beigey coloured space here. The vacuole will be pushing outwards, which is represented by these black arrows, and the cell will be nice and firm. We say it's turgid. So this turgor pressure is what holds very young plants up if they haven't got any wood, and the turgor pressure keeps the cells firm. We say that the cell is turgid, full of water, a turgid cell. This is what stops plants from wilting. If plant cells lose water because it moves outwards by osmosis, so for example, if a plant cell was put into a strong sugar solution or a strong salt solution, then the contents of the plant cell would be more dilute. There would be more water molecules inside the cell, and water would move out of the cell by osmosis. This would mean that the vacuole would shrink. There wouldn't be the pressure pushing outwards on the cell wall, and the cell will go floppy. We say it's flaccid. This is what happens when plants wilt. If water continues to move out, eventually the contents of the cell will be pulled away from the cell wall, and we say that the cell has become plasmolysed. So you can see that plant cells are protected somewhat because they have this rigid cell wall to provide strength, but all cells can be damaged if water moves out of them by osmosis. In the case of animal cells, if water was moving into animal cells, for example, our blood cells, they would swell up, but because they don't have a cell wall, animal cells would burst. In other words, if we took some red blood cells and put them into some pure water, the cells would swell up, filling up with water by osmosis, and eventually they would burst.